This slide illustrates the two most recent duplications in the ABP gene region. These occurred as duplications of blocks of multiple ABP genes that probably occurred by non-allelic homologous recombination, or NAHR. The block at the top, underlined in black, is the group of genes that gave rise to the penultimate duplication, resulting in two blocks in the center, one underlined in green and the other in orange. At the junction of the two daughter blocks is a beta-gamma subgene that is crosshatched to show that it was lost during the duplication. The right-hand daughter block of genes had a central region designated in the figure as XYZ that gave rise to the ultimate duplication shown below as two blocks, one identified by underlining in orange and the other by underlining in blue. Together, these duplications resulted in the final product at the bottom, a segment that represents the central 50% of the entire ABP gene region. We contrasted the repeat density in 50 kb non-overlapping bins in the ABP gene family regions and in flanking regions 1 megabase proximal and distal to the gene regions. L1-MDT repeat families were found to be high in densities in the ABP gene regions. The densities for the L1 repeats were more than 10 times higher in the ABP regions than the flanking regions, and this family covered on average more than 30% of the 50 kb non-overlapping bins from the ABP region in both genomes. Abrupt transitions at the gene region boundaries are striking, and they suggest that their higher densities are tightly associated with ABP gene duplications in both the mouse and rat. Our paper provides the explanation by which we discounted a similar role for the ERV2 elements, also found in high densities in these gene regions. We also observed that the major accumulation of L1 elements occurred after the split of the mouse and rat lineages, and that there is a striking overlap between the timing of L1-MDT accumulation and expansion of the ABP gene family in the mouse genome. This slide shows the breakpoints in the L1-MDT elements, flanking the most recent ABP gene duplications in the mouse genome. The three bars in panel A show the L1-MDT elements on the left flank, in the center, and on the right flank of the duplicated segments in the last block duplication by NAHR. The vertical arrows above and below the L1-MDT elements is the breakpoint, shown in detail in panel B. Sequence 1 has additional sequence in the break not shared by sequences 2 and 3. This region contains two interior duplications, a larger one bracketed in red and a smaller one bracketed in green. The details of the events in this breakpoint are discussed in our paper. Panel C shows the consequences of NAHR at this breakpoint for SNPs in the duplicated gene region. These patterns are consistent at 384 divergent sites proximal and 127 divergent sites distal to the mispaired AGCA/GGTT shown in the gap, and they support the idea that this is the breakpoint for the duplication.